welcome to the new topic in study medicine. So today we will be discussing transport process in general physiology. Okay, transport processes. So let's begin. Basically, there are two types of transport processes. Okay, the first type, across the membrane. Across the membrane, the transport happens across the membrane, and another way is through the membrane. Okay, now in case of across the membrane, there are two types: endocytosis, and exocytosis. And in case of through the membrane, there are three types. You guys have heard about it: osmosis, diffusion, and last but not the least, active transport. Okay, so let's discuss it one by one. Okay, so first we will be discussing about across membrane. Across the membrane, the transport happens. Okay, in this particular case, mostly a vesicle will be formed in the membrane. Okay, and then the thing gets transported from membrane inside. Okay, so here vesicle transport happens. What vesicle is being formed. Okay, now across the membrane, there are two types of across the membrane. One is endocytosis. Now endocytosis, basically it is occurs in large molecule particles, just like foreign substance. Large particle particles. Okay, and endocytosis can be divided, or there are two types of endocytosis. One is phagocytosis. And next one is pinocytosis. Okay, pinocytosis. So what are the difference? So phagocytosis, we can also call it as cell eating. In terms of pinocytosis, we can call it as cell drinking. Okay, so in this particular phagocytosis, the example, you can take an example like bacteria entry in the neutrophil. Okay, entry in neutrophil. And in pinocytosis or cell drinking, you can think about an example of soluble proteins that enters into the cell. Okay, soluble protein enters into cell. So we discussed about endocytosis. Okay. Now we will discuss about exocytosis. Exocytosis. So it's basically also called as reversed pinocytosis. Reversed pinocytosis. And exocytosis can be divided into, again, there are two types of exocytosis. One is constitutive exocytosis. Constitutive exocytosis, and other is regulated exocytosis. Now what's the difference? Constitutive exocytosis, basically it's a continuous process, and regulated, it's being regulated, it doesn't happen continuously. Like consecutive example for consecutive exocytosis is like mucus secretion. Okay, mucus secretion. And regulated exocytosis, example could be hormone secretion. And regulation happen, then only hormone is being secreted. Okay, so these were the two things. What across the membrane? The across the membrane has been discussed. Now we talk about through the membrane. Okay, so the first thing which we will talk about through the membrane is osmosis. So basically, what is osmosis? Water moves from low solute to high solute, or high solute concentration. So low solute concentration to high solute concentration. Water moves. Okay. Now, as there's one important phenomena which you can see here. It's also called as solvent drag protein or bulk flow. Or solvent drag protein. Why? The reason is simple. When water moves, as it moves in bulk quantity, along with it some amount of solutes also moves. Okay, so small amount of solutes also moves when water moves in a bulk quantity. That's why it's called a solvent drag portion, because the water drags the solvents along with it. So now the best example for this particular thing is you can see a descending loop of a descending limb of Henle's loop. Descending limb of Henle's loop. Okay, it's the best example you can think about. Now, as we have taken the topic of osmosis, just let us understand the difference between osmolarity and osmolality. Okay, so what are the difference? So basically, in osmolarity, it's number of solutes by solution in liters. Okay, but in case of osmolality, it's mass of solute by kilogram of water. So another thing which you have to keep it in mind, that in case of temperature, it will influence osmolarity. Okay, it will influence osmolarity but does not influence osmolality. So this is the thing which you have to keep it in mind. Just okay. So osmosis, we discussed osmosis. Now let's just discuss about diffusion. Diffusion. Okay, so first thing which we have to keep it in mind, that diffusion can be divided into simple diffusion and facilitated diffusion. So what is diffusion? First of all, here in this diffusion, no ATP is used. Okay, so as no ATP is used, it is passive transport mechanism, and here the net effect is downhill transport. Okay, downhill transport happens. Basically, what does downhill means? From high to low concentration it moves. Okay, now let's the types. Okay, simple diffusion. Simple diffusion. So in this particular simple diffusion, it's the same. No ATP is being used. It's a passive transport, downhill transport. Okay, and basically here kinetic energy drives the diffusion. And the things which we have to keep it in mind about is that the factors that can affect simple diffusion. Okay, the first one is lipid solubility. So if the solubility of lipid increases, it can move or get transported. Like the lipid soluble, think about carbon dioxide. It is more soluble than oxygen. Why? So carbon dioxide is more soluble than oxygen. Then let's go talk about the second factor which takes in simple diffusion, that is number of channels. It's not sodium, okay, it's number of channels. So as number of channel increases, the transport simple diffusion also increases. Like example, the best thing is that potassium channels is more compared to the sodium channels, so potassium moves, simple diffusion happens more compared to sodium in such a case. Okay, then the third factor is temperature. So temperature, it is directly proportional. Okay, so the temperature is directly proportional to simple diffusion. As temperature increases, diffusion will also increase. Then the fourth one is surface area. It's also directly proportional to simple diffusion. Then comes the fifth one, thickness of the membrane. Thickness of the membrane. And in this particular case, it is inversely proportional. So as the thickness of the membrane increases, simple diffusion will decrease. Then the sixth, size or radius of the substance. Just think about the radius or size of substance. So it's also inversely proportional. As size increases, it will be difficult for it to go through simple diffusion. Then 7, concentration gradient. It's directly proportional to the concentration gradient. Then comes, like, basically means if the concentration gradient here is higher outside, so it will move inside pretty quickly, or move pretty quickly. Okay, now eighth one, that is pressure gradient. Then the ninth one, it is electrochemical gradient. Okay, so these are the things. Electrochemical gradient, you can understand, is like we talked about that sodium leaky channel, right? Just think about in that particular case, as the sodium concentration is higher, it acts as a cation, so it's positive in charge, and as sodium concentration is less inside, so cation charge is less comparatively. So as the positive cation charge is higher outside, it will help to move inside by simple diffusion. Okay, so these were the things, factors which can affect simple diffusion. Now we will talk about facilitated diffusion. Okay, facilitated diffusion. So in this facilitated diffusion, ATP is not used, no ATP, and it's also a downhill transport. But in this particular thing, there's a carrier, and facilitated diffusion is a carrier mediated transport. Okay, carrier mediated transport. So in facilitated diffusion we discussed these are the only things. Now let's discuss about the last thing, active transport. Okay, active transport. So in active transport, ATP is used, and as ATP is being used, it does uphill transport is happened most of the time. Uphill transport basically means not from high to low concentration, but from low concentration to the high concentration, low to high concentration. Now the active transport can be divided, again classified into two: primary active transport and secondary active transport. So let's talk about primary active transport. In this particular thing, ATP is used directly and carried by a pump. Okay, carried by a pump. But in case of secondary, here also ATP is used, but it is used indirectly. Why it is being used indirectly? Because here a carrier is being present, because of which the ATP is used indirectly. So a carrier is present in secondary active transport. So the best example for secondary active transport, we discussed about glucose transport in GIT and kidney. Glucose transport in GIT and kidney, and it is done by SGLT, not GLUT. It is being done by SGLT. Okay, and in case of example for primary active transport, you can just think about sodium potassium pump. Sodium potassium pump. Okay. So these were the things which you have to keep it in mind when you think about transport process. Okay, now this extra thing which you have to just keep it in, just note it down. It's about sodium potassium pump. Okay, sodium potassium pump. Okay, so basically it is, as we discussed, it is primary active transport, and it has 3 sub units and 5 binding sites. So let me draw a pic of sodium potassium pump. So subunits, basically we drew it. Okay, now two is potassium binding site, and 5 it is pain binding site. And about 1 it is sodium binding site. 3 ATP binding site, and 4 it is phosphorylation site. Relation site. Okay, so this was the sodium potassium pump. Now, as we discussed, let's discuss about the GLUT protein. So there are different types of glucose transport proteins. There are different types. Okay, so let's discuss one by one. GLUT1, it has been expressed constitutively expressed in ubiquitous. Okay, then GLUT2, pancreatic cell, and the glucose leaves or exits the cell by GLUT2. Glucose exit cell by GLUT2. Then comes GLUT3. GLUT3, it is also present in brain. Then comes GLUT4. It is insulin dependent glucose transporter, present basically in muscle and fat. Okay, and GLUT5, it is basically present in fructose transporter. So these are the things which you have to just note it down and just keep an eye. So thanks for today's session. Hope we can see you in the next session. Bye.